Good morning, students. Today we are again continuing the digestive system, in which the organ small intestine, its function of digestion and absorption. The small intestine, its function of digestion and absorption, that is the assimilation of the food. So the digestive tract, that is the alimentary canal or the gastrointestinal canal, is having different parts, and these parts are called digestive organs. The first one is the buccal cavity, second is pharynx, then the third one is esophagus, fourth is stomach, then fifth is intestine. So in the intestine is also a part of the digestive system. Now coming to the intestine, small intestine is the part of alimentary canal which extends from the pyloric end of the stomach to cecum. The small intestine is the part of alimentary canal which extends from the pyloric end of the stomach to cecum, the first part of the large intestine, that is the cecum. It extends from the pyloric end of stomach up to the cecum, which is the first part of the big intestine or the large intestine. The small intestine consists of three parts. It consists of three parts. They are, first one is duodenum, second jejunum, third one is ileum. Duodenum, jejunum and ileum are the three parts of the intestine. Coming to the duodenum, it is C-shaped fixed part which is attached to posterior abdominal wall by peritoneum. It is a U shape or C shape. U or C, like this. It is like this. So U shaped or C shaped part which is fixed anteriorly, which is attached to posterior abdominal wall. It is attached to the posterior abdominal wall by peritoneal membrane. The head of the pancreas lies in the concavity of the duodenum. The head of the pancreas lies in the concavity of the duodenum. In the U shape, in the U groove of the intestine, that is the duodenum, the pancreas lies. Also, the bile duct and pancreatic duct open together at the concave surface. So at this surface of the duodenum, the bile duct and the pancreatic duct, they together open and they open at the duodenal level. A small immense at the opening is called ampulla of Vater, Vater, Vater. Ampulla of Vater is present on the duodenum. Now coming to the jejunum, the second part, jejunum, it is the continuation of duodenum, and it is middle portion of the small intestine. It is the middle portion of the small intestine which is the continuation of the duodenum. Then it forms the last part of the small intestine, that is nothing but the ileum. Ileum is the last part of the small intestine. So duodenum, jejunum and ileum are the three parts of the the small intestine. Now coming to the structure of the intestine. Small intestine consists of the same four coats, four layers which are present in the stomach also. So I have explained in the stomach. The four coats. The first one is the peritoneal coat made up of serous membrane. The second is the muscular coat made up of circular and longitudinal muscle fibers. Third one is the submucosa coat made up of areolar tissue. And the fourth is mucus coat, which is the inner lining. So the inner lining is made up of mucus. The mucus coat is in the intestine. The mucus coat plays a very important role. The mucus coat, the mucous membrane of the intestine, is thrown into many folds of the many folds called pilica circularis, plicae circulari, which are the folds of the mucous membrane, and they are also called as villi, which absorb carbohydrates, proteins and fats. Villi, they are called as villi. The folds or the finger-like projections of the mucous membrane are called villi, villi, very important structures of the small intestine. The inner layer, that is the mucus coat, is thrown into many folds, and they are just like finger-like structures. They are finger-like structures like this, and these finger-like structures are called villi. The villi. Digestion in small intestine. The acidic chyme from the stomach enters into the duodenum. The acidic chyme, the chyme enters from the duodenum into the acidic chyme from the stomach enters the duodenum. There it mixes with the alkaline intestinal juice called succus entericus. The alkaline intestinal juice called succus entericus. Succus entericus is nothing but the alkaline intestinal juice. Intestinal juice is called succus entericus, which is alkaline in nature. The intestinal juice succus entericus is alkaline. And the alkaline secretion from the liver and pancreas. The alkaline secretions from the liver and the pancreas also come in the intestinal juice. In the small intestine, digestion is carried out by the following enzymes of intestinal juice. The first one is enterokinase, which converts trypsinogen of pancreatic juice into the trypsin. The trypsinogen present in the pancreatic juice is converted by enterokinase, the first enzyme, into trypsin. So the trypsin is formed by the enterokinase by converting the trypsinogen of the pancreatic juice. Now the second one is erepsin, 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 which converts polypeptides into amino acids. The polypeptides are converted into amino acids by the action of erepsin enzyme of the intestinal juice. Next, third one, sucrase, maltase and lactase. These are all the carbohydrate enzymes. Sucrase, maltase and lactase, which convert the corresponding disaccharides into monosaccharides or the glucose. The disaccharides like maltose, they convert the maltose into glucose. So the final structures, amino acids of the protein, glucose of the carbohydrates are formed in the intestine. Then the absorption in small intestine is by the absorption of digested food occurs in the small intestine through villi. The absorption takes place in the villi. Villi are minute projections which are present in the inner mucous coat of the intestine. I have already told they are finger-like structures of the mucous coat. The villi give a velvety appearance to the intestinal mucous membrane. Each villi has a central lymphatic vessel called lacteal. Fats are absorbed into lacteals and carried to thoracic duct. Second, the network of capillary surrounding the lacteals. Digested products of carbohydrates and proteins are absorbed into these capillaries. They are carried to liver by portal veins. Third one, lymphoid tissue which holds together the the lacteals and capillaries. The lymphoid tissue is also present in the villi. The villi contains the finger-like structure. It contains the lymphatic vessels called lacteals and network of capillaries and the lymphoid tissue. So three important parts are present in the villi. The lymphatic vessel, lacteal, absorbs fats in the form of fatty acid and glycerol and lacteals and carried to the thoracic duct. Then the network of capillaries surrounding the lacteals, they absorb digested products of carbohydrates and proteins and carried to the liver by portal vein. Then lymphoid tissue holds together the lacteals and the capillaries. It gives support to the lacteals and the capillaries. So this is about the small intestine. Coming to the large intestine, intestine and its functions. The large intestine colon extends from the end of ileum to rectum. Large intestine consists of the following parts, that is the cecum, the first one, appendix, the second one, ascending colon, transverse colon and sigmoidal colon. So these are the different parts present in the large intestine. The large intestine is divided into first one, cecum. The first is cecum. It is a short rounded sac which lies in the right iliac fossa. It commences at ileocecal wall where the ileum joins the cecum. It is a short rounded sac-like structure lying at the right side of the iliac fossa. Second one, vermiform appendix. Appendix is vermiform, and this appendix is it springs out from the cecum at about an inch from the ileocecal junction. It is present in the right iliac fossa. The lumen of appendix communicates with that of cecum. The appendix is composed of the same four coats as intestine, but the submucous coat contains lymphoid tissue and it is vestigial in the human beings. The appendix is vestigial in the human being. The function of the vestigial is not found out in the human beings. Third one is the ascending colon. It ascends upwards from cecum and in front of right kidney. It turns to left below the liver and forms the transverse colon. Fourth one is transverse colon. It is the loop of large intestine which extends between the lower surface of liver and spleen. At the lower surface of spleen, it turns downwards to form the descending colon. Coming to the descending colon, it extends from the lower surface of spleen to brim of pelvis. It lies in the left lumbar region. The last one, sixth, is sigmoid colon. It is a continuation of descending colon and it continues below with rectum. It is continued below with the rectum. Structure of the large intestine. The large intestine has the same four coats, that is peritoneal, muscular, submucosa and mucosa layers. But the only difference is longitudinal muscles are arranged in three bundles. The mucous membrane doesn't have villi. The villi are absent in the in the large intestine. In the small intestine, the villi are present. The mucous membrane thrown into small folds and they are called villi, the finger-like structures. But here the villi are completely absent in the large intestine. Functions of large intestine. Digestion. This is carried out by the microorganisms of the colon. They act on the undigested and unabsorbed residues from small intestine. They act on the undigested and unabsorbed residue food materials. Then absorption. All carbohydrates, proteins and fats are already absorbed in the small intestine. Only water and glucose are absorbed in the colon. The water and the absorbed excess of water and the excess of glucose is present in the large intestine. If it is present, then it will be absorbed by the large intestine. Coming to the secretions, mucin is the only secretion. It lubricates the colon and facilitates the passage of fecal matter. Mucin is secreted by the large intestine and it is the only secretion which lubricates the large intestine and facilitates the passage of undigested food material. Then excretion, the fourth one is the excretion. Iron and some purgatives are excreted in large intestine. Iron and some purgatives are excreted in large intestine. Coming to the next one, that is the rectum, anus and defecation. Rectum and anus. Rectum, it occupies the lower posterior part of the pelvis. It extends between sigmoid colon and the anus. The lower part of the rectum is dilated and it is called rectal ampulla. Rectum occupies the lower posterior part of the pelvis, extends between sigmoid colon and anus. Now coming to the anus, it is small canal measuring about 1 inch in length. The opening of anus is guarded by sphincter called anal sphincter. This sphincter is under voluntary control. There is a sphincter called anal sphincter is present in the anal region, and anus is a small canal measuring about 1 inch in length. It is 1 inch in length and the opening is guarded by the anal sphincter. So this is about the different parts of the digestive system. The digestive system contains buccal cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum, cecum, rectum and anus. So these are all the parts of the digestive system, and the digestion of the materials, food materials which is ingested is also explained, and different enzymes produced by the stomach, intestine and buccal cavity are also discussed. And this is about the digestive system. And there is one more thing, the digestive system is supplemented by the glands. They are nothing but liver, and pancreas. We have to discuss about the glands. Now, coming to the next point, digestion of food in the alimentary canal. The digestion of the food in the alimentary canal. Food contains carbohydrates, proteins, third, fats. All these constituents of the food are digested in the alimentary canal as follows. Carbohydrates, proteins and fats are the three constituents of the food material, and they are digested in the alimentary canal. First one, the carbohydrate. Toilin salivary amylase present in the saliva converts cooked starch in food into sugar called maltose in the buccal cavity. The carbohydrates digestion starts in the buccal cavity itself, the first part of the buccal cavity, wherein the salivary amylase or the toilin enzyme secreted by the salivary glands converts the cooked starch into maltose sugar. The conversion occurs in the mouth, that is the buccal cavity. All sugars are converted to simple monosaccharides like glucose by the action of enzymes sucrase, maltase, lactase in the small intestine. Glucose is absorbed through the capillaries of the villi in the small intestine. It is then carried to the liver by portal vein where it is stored as glycogen. So, all the carbohydrates are completely digested or converted into simple monosaccharides in the small intestine by the action of sucrase, maltase, lactase enzymes of the intestinal juice, and then this glucose is formed and glucose is taken up to the liver by the portal vein where it is stored in the form of glycogen. Coming to the proteins, the digestive enzymes pepsin of the stomach, trypsin and erepsin of small intestine convert the proteins into peptones, polypeptides and finally into amino acids. The amino acids are absorbed through the villi of small intestine and carried to liver. Coming to the fats, lipase enzyme of pancreas which is poured into the small intestine converts fats into fatty acid and glycerol. These two products are absorbed through lacteals of the villi. They are carried to the thoracic duct through cisternae chali. From the thoracic duct, they enter into the blood through left brachiocephalic vein, that is the left subclavian vein. So this is about the digestion of food in the alimentary canal, that is the carbohydrates, proteins and fats digestion in the alimentary canal. Coming to the absorption of the food, it's already explained. Mouth, absorption of the food does not occur in the mouth. Only drugs like nitroglycerin and isoprenaline are absorbed through buccal mucosa. Stomach-nally, no absorption of food occurs in the stomach except glucose, but water and alcohol can be absorbed in the stomach. Then coming to the small intestine, products of carbohydrates digestion takes glucose, carbohydrates digestion like glucose and other simple sugars occur through capillaries of villi. Products of protein digestion like amino acids are also absorbed through capillaries of villi. Products of fats metabolism like fatty acid and glycerol are absorbed through the lacteals of the villi. And in the large intestine, water and glucose and certain salts are absorbed through the mucous membrane of the large intestine. So this is the absorption of the food. in the alimentary canal. Now, the accessory organs of the digestion are liver and bile ducts and pancreas. I have already told there is the alimentary canal, which is the intestine gastrointestinal canal, alimentary canal, and some glands are there. They are nothing but they are included in the digestive system. They help in the digestive system. The glands which help in the digestive system are liver, bile duct, and pancreas. Liver is a large gland which secretes the bile, and it is carried by bile ducts. Then pancreas also secretes the enzymes which augment the digestion of the constituents of the food materials. Now coming to the liver first. Liver is the largest abdominal organ. It lies in the upper part of the abdominal cavity below the diaphragm and under the cover of lower ribs. It is the largest abdominal organ. It lies in the upper part of the abdominal cavity. Coming to the external features, externally the liver contains 2 lobes and 4 surfaces. Lobes are right lobe and the left lobe. Surfaces are superior surface, which is in contact with the under surface of the diaphragm. Inferior surface, which is facing the abdominal viscera. The hilum or portal fissures is present in the inferior surface. The blood vessels of liver and bile duct pass through the hilum. Hilum is present in the inferior surface. Coming to the anterior surface, the third one, which is separated from ribs and costal cartilages by the diaphragm. Then the fourth one is the posterior surface, which lies in front of the vertebral column, aorta, inferior vena cava, and lower end of the esophagus. So there are 4 surfaces present in the liver, and there are 2 lobes, right and the left 2 lobes are present. The microscopic structure of the liver. The liver consists of large number of liver cells called lobules. Lobules are the liver cells. They are large in number present in the liver. Each lobule has a central vein or interlobular vein. The connective tissue lying in between the lobules contains portal vein, hepatic artery, bile duct. So the connective tissue lying in between the lobules, it contains portal veins, hepatic artery, and the bile duct. Blood supply to the liver. Blood is brought to the liver by hepatic artery, then portal vein, but blood is carried from the liver to inferior vena cava through hepatic veins. Coming to the hepatic artery, it supplies oxygenated blood to liver. It is a branch of celiac fluxus, which in turn arises from abdominal aorta. Celiac flux arises from the abdominal aorta, branch of the abdominal aorta, and supplies the oxygenated blood to the liver. Coming to the portal vein, second one, portal vein also brings blood to liver. It carries blood from stomach, spleen, and intestine, which blood contains nutrients. This blood contains nutrients. The portal vein brings blood from liver, carries blood from stomach, spleen, and intestine, which contains the nutrients like amino acids, glucose, and glucose. So carbohydrates and proteins and carbohydrates. So these nutrients are brought to the liver by the portal vein. It divides into interlobular veins which lie in between the lobule of the liver. They subdivide and ultimately form central veins. One central vein lies in the center of each lobule. Hepatic veins. Next are the hepatic veins. They carry the impure blood of the liver and drain into inferior vena cava. The central veins of each lobule unite to form sublobular veins. The sublobular veins unite to form several hepatic veins. The hepatic veins join with inferior vena cava. So, hepatic veins are there, portal veins are there, hepatic artery is there. Hepatic artery supplies the oxygenated blood to the liver. Then hepatic veins carry the impure blood to the superior inferior vena cava. Coming to the bile duct, the secretion of the liver, that is the bile, is the secretion of the liver, is carried through bile duct, which are formed by the union of biliary canaliculi. The biliary canaliculi are small biliary channels present in between the lobules of the liver. The bile duct from the right and the left lobes of the liver unite to form common hepatic duct. The hepatic duct unites with cystic duct of gallbladder to form the common bile duct. Later, the common bile duct unites with pancreatic duct in the duodenum at the papillae called ampullae of Vater. Functions of liver. Coming to the functions of liver, secretion of bile, synthesis and storage of glycogen, formation of urea by the deamination of amino acids, synthesis of plasma protein like albumin and globulin, conversion of unsaturated fats into saturated fats. Then storage of iron and vitamin B12 which are necessary for the formation of RBC, synthesis of prothrombin, prothrombin and fibrinogen which are necessary for blood coagulation, then synthesis of heparin, the neutral anticoagulant. The ninth one is the production of the heat as a result of metabolic reaction. 10th one is inactivation of toxic substances and drugs. Then the last 11th one is storage of vitamins A, D, E and K. So liver has about 11 functions. It is a very big gland of digestion and it has very important functions. The functions of liver are very important and you please go through the functions of the liver from the notes which I have given. Tomorrow we will take up the gallbladder and the pancreas. That completes the digestive system. So tomorrow we will go for the gallbladder and pancreas. Thank you very much.